Hi everyone, this is Caroline with Fiveable. Thanks for tuning in tonight. Hopefully you're staying warm wherever you are. I'm in Chicago, so it's like negative 30 degrees right now, but we didn't have school, so that was the nice part about that. But wherever you are, I hope you're staying warm and staying safe and all that jazz, because this weather is no joke. So anyway, thank you for tuning in. Tonight we are talking about bacteria and viruses. This is my very favorite topic. In college, I was a microbiology major, so this is like what I spent all of my days studying, and so I'm really excited to share that with you. So just a quick announcement. So obviously this session is free, but all replays and all materials from sessions and worksheets and PowerPoints and all of that jazz come along with the premium membership, which is $20, I believe up through April. That's what I'm double checking for you guys right now, but it's $20 to enroll. You can think about it like going out to dinner, but instead you get lots of AP prep for every single class. So if you're in multiple AP classes, that's really awesome. And you can get credit for all of them. That's until April 1st, and then it's $35 after April 1st and between April 1st and exams. And again, it's for replays, live stream notes, linked slides and resources, and extra cram review that will happen in May. So might as well sign up now while you still can for $20 as opposed to $35. And then you get to see more AP Bio and lots of other content as well. All right, so we're going to jump in. Sorry, just sharing my screen with you. Ooh, why? Why do bad things always happen during these presentations? Always, always. Okay, here we go. Once it decides to load, even though it's been loaded for 30 minutes for some reason. There's always problems. All right. So bacteria and viruses, again, my very favorite thing ever in the whole world to talk about. So I'm excited to share some of my favorite things about bacteria and viruses with you as we move through. So the purpose here, why do you need to know about bacteria and viruses? One, they're really cool, in my humble opinion. But two, you need to know the major differences between bacteria and viruses for the AP exam. With bacteria, we talk about antibiotic resistance all the time. It's a perfect example of evolution. It talks about cells. It talks about gene transfer, DNA, all sorts of good things. So antibiotic resistance is really important to talk about. This will also help us to better understand the immune system, which we'll be covering shortly here. And I think about 2 weeks. But if you don't know about the things that are attacking or trying to circumvent our immune system, then it's hard to understand how our immune system is combating those things. Okay, so that's our purpose for tonight. Pretty cool stuff and fitting because it's sickness season. The flu is upon us and lots of other things as well. Okay, so we're going to start by talking about bacteria. You're probably pretty familiar with them, I'm sure. I mean, we talked about them in our cells unit. You know about prokaryotes, and you've probably learned about them in the past. So bacteria, they're prokaryotic, as I said, unicellular, and all bacteria have DNA as their genetic material and ribosomes, right? So classic example of a prokaryote with DNA and ribosomes. So the reason that I'm going into detail about this, while you probably already know it, is because this is going to be different for viruses, and so it's important to know what bacteria have in order to compare that with what a virus has. Second key fact about bacteria is that they have all characteristics of life. Okay, and what I mean by that is they can reproduce on their own, they can produce their own energy, they can adjust to their environment, they can change over time, they can replicate, all sorts of things. Believe it or not, scientists have like a checklist that they go through to see if something is alive or not. So bacteria are considered alive, which again, I'm sure is no surprise to you. But again, we're comparing viruses here, and spoiler alert, viruses are technically not considered living things, which is pretty crazy. So it's important to know that bacteria are. Another fun fact about bacteria is that they have the greatest percentage of biomass on Earth. So what does that mean? So biomass would be if we take all living things on Earth and we weigh them, right? And we have like the entire mass of living things on Earth. Then we can segment that into percentages of animals, plants, marine life, humans, whatever we want to do. We can take the mass of just those components and figure out what percent of the total mass of living things on Earth that is. And this is mass, right? Not like quantity, but weight, if you will. So bacteria, little teeny tiny, essentially like massless things, actually take up the most percent of our biomass, right? So there's more mass of bacteria on Earth than of any other living thing, which is super crazy and super awesome. There are actually like 10 times as many bacterial cells inside of you as there are human cells inside of you. So you are more foreign than human, and that's because the entirety of our digestive tract is lined with bacteria, as well as our skin is coated in bacteria, our teeth, etc. But yeah, so there's actually 10 times more bacteria inside of you than actual human cells, which is pretty crazy. And the vast majority of bacteria are incredibly beneficial to us and have really positive things for us. It's actually less than 1% that cause disease, but they give everybody else a bad rap. So that's our bacteria. Here's the basic structure of a bacteria. So I'll get my little pointer out. So you'll notice a couple things. So we have a cell wall, which is common of prokaryotes, right? And a cell membrane, again, common of prokaryotes with ribosomes and DNA. A couple fun things, flagellum, which is just singular for flagella, allow bacteria to have movement so that they can travel through different spaces. Pili are these little stick-like things, and their role for a bacteria is to stick to surfaces. And so if you think about it, in order for a bacteria to really be successful, they have to get somewhere, stick there, and stay there in order to start to replicate, right? But then if you think about humans, everything about us is trying to get rid of bacteria. So when you're sick, you're sneezing, you're blowing your nose, you're coughing, all of those things are responses in order to rid your body of bacteria. So in order for bacteria to actually colonize inside of you and make you sick, they have to be able to stick somewhere and reproduce without you coughing them up, sneezing them out, or having a white blood cell or something else eat them. So these pili really help with them sticking to surfaces so that they can replicate before they've been detected or excreted in some way. Another thing to note, you'll see this little red layer here, and that's peptidoglycan, and that's a special compound that exists in bacterial walls that actually is one of the big things that we target with antibiotics because it's something that, again, is unique just to bacteria, and so they are susceptible to this, whereas we would not be because we do not have peptidoglycan in our cell walls. It makes them stronger and more rigid. We don't have cell walls, but makes them stronger and more rigid, etc. And the thickness of that peptidoglycan gives us gram-positive and gram-negative bacteria, if you've heard of those before. So just like a structural difference between types of bacteria and the thickness of their cell wall. All right, going along with bacterial structure, bacteria come in a couple different shapes and sizes. So they can be spherical, rod-shaped, or spiral, and that actually gives them their name oftentimes as well. So Streptococcus pneumonia is what causes strep throat or pneumonia infections, and that's because the bacteria are spherical cocci. Tetanus is caused by bacilli bacteria, and they are named as such. Spirilla is less common, but most of the time we'll have rod-shaped or spherical. And then within that, we can have some variety, like sometimes with spherical, they'll always grow in chains or in pairs or in singular units. But it allows us to determine a lot about the bacteria based on the shape. You don't need to memorize those shapes for the AP exam, just giving you some background knowledge of how scientists identify different bacteria. Okay, so the uses. As I said, less than 1% of bacteria are actually going to be harmful to us, and a lot are actually helpful. So I'll just name a few ways. Sometimes this comes up on the AP exam. It might be in a free response or in the meat of some sort of multiple choice question. Just kind of interesting to hear about. So one, they help make food interesting. So they're going to help us to create lots of different flavors and textures in our foods. Things that are pickled, like pickles or sauerkraut, involve some fermentation from bacteria. Yogurt is full of good bacteria. If you remember, like back in the day, the Jamie Lee Curtis Activia commercials, she talks about all of the probiotics that exist in Activia yogurt, but all yogurt actually has natural live occurring bacteria in it, which is great for you and good to help your digestive system gain back some bacteria, especially if you've lost some. Maybe you took an antibiotic and that wipes out some of the bacteria inside of your intestines. Yogurt is a good way to replenish that. Cheese, Swiss cheese, if you look at the holes in Swiss cheese, those are propionic acid, which is a production of a bacteria. It's actually the same bacteria that gives you pimples, weirdly enough. And then, yeah, other things like that. So making food interesting, we love that. They also help decompose organic matter. So we actually use them in sewage systems all the time to help break down our solid waste, which is not a job that any person would want to do. So thank you, bacteria, for breaking down our solid waste. Nitrogen fixation, so we'll talk about this more in ecology, but plants are not able to convert nitrogen gas into usable organic materials or ammonia or something that they can use, and nitrogen is going to be one of the most important elements that we have. Carbon, hydrogen, oxygen, and nitrogen make up the majority of our macromolecules, and so bacteria, actually, we rely on bacteria to convert nitrogen gas into usable like ammonia and other nitrogen containing compounds so that plants can incorporate them, and then we have a source of nitrogen. But without bacteria, we would be incapable of doing this. More on that when we get into ecology in a month or two. Human health, I've already alluded to this, but bacteria are really, really helpful. So our skin is coated in them. And that's a really good thing because if other bacteria land on our skin or get on our skin, our natural bacteria will fight them off, right, and outcompete them so that we don't get skin infections or other types of infection from those bad bacteria. So our natural flora is what it's called, is able to kind of fight off and outcompete any other competing bacteria. Now, good bacteria can be bad in certain circumstances. If you get a cut or something like that and some of your normal bacteria gets inside, it can lead to infections and problems. But for the most part, they're out there to help us. And then again, they're in our gut digesting things and helping us make vitamins all the time. And we talked about the lac and the trip operon with gene regulation. So they help us break down lactose. They help us produce tryptophan. Really important amino acid. They do lots and lots of great things for us. We would not be able to survive without them. The last one might be my favorite one. So biotechnology. So bacteria contribute in a big way to our existence and our future in medicine. So we use them to obviously help make us antibiotics so that we can defeat bacterial infections. But we also use them all the time. And this is going to be a big focus for next week's biotechnology lecture. We use them all the time to produce useful proteins because if you remember from when we were talking about transcription and translation, all living things transcribe and translate in the same way. We all have DNA, we all have the same base pairs, we all have the same amino acids. So what we can do is we can take a gene of interest, a human gene of interest. For example, the gene that produces the protein insulin, which is necessary for regulating blood sugar and is in low supply in someone who has diabetes. We can take that gene out of the human genome and actually implant it into a bacterial genome, and that bacteria will then go ahead and transcribe and translate that gene for us to produce that protein, in this case insulin, and then we can purify that insulin and use it for injections and things like that for people who need it because they can't produce it on their own. And so we actually use bacteria quite frequently in this way, where we can kind of manipulate their genome, have them make proteins that are useful to us, and then capitalize on that. So a lot of medical advances that we have. Same goes for human growth hormone. Growth shots if kids are underdeveloped or have some sort of inhibition with their growth, we can help make that hormone. Lots of great medical advances have come from using bacteria in good ways. So it's important to remember those. The AP exam also likes to write about them sometimes, so it's good things to know about. All right. But then obviously we have the bad ones. And again, as I said, this accounts for less than 1% of all bacterial species, but they are the loudest of them. And so how do they work? There's two major ways that bacteria are going to harm us. One is by using our cells for food. Obviously that's bad, right? If they're eating our cells or breaking down our cells, we're going to be affected by that. And that does happen sometimes in our bodies in various places. And it can be something major like flesh-eating bacteria that truly just like eats through your flesh and your muscle and your tissues. Or it could just be that they're rupturing cells, like in strep throat. They're rupturing cells in the back of your throat, which is causing irritation and inflammation. But more common probably is that bacteria release toxins. And toxins are toxic proteins that can interfere with daily actions and kill cells. So I kind of have an example here, but they can kind of hijack our cells or stop normal like protein synthesis or something like that as well. And those two ways are going to be kind of how bacteria can kind of damage us, but at the same time, through this damage, they're also leading to alerting us of their presence. And so we have T cells and B cells and white blood cells and all sorts of things that we'll talk about in about 2 weeks that are then alerted to inflammation and any sign of toxin release so that we can circumvent the attack. All right, so some examples of diseases caused by pathogenic bacteria: food poisoning, tuberculosis, syphilis, strep throat, scarlet fever, whooping cough, bacterial meningitis, pneumonia, tetanus, bronchitis. And so bronchitis is like a really mucousy, deep cough caused by infection of the bronchioles, which are like little tree branches that exist in our lungs, which is the pathway for air in and out. Couple other ones to mention. So strep throat you may have had before. I used to get it. I had it a few times when I was growing up, where you have these little white patches, which are truly bacterial colonies that are forming on your tonsils and on your tongue, and you get really red and inflamed again from the toxins and from cells rupturing on your tonsils and in your throat. Bacterial meningitis, but again, you'll notice on this list, and you might already be very well familiar with this, like I don't have the common cold or just like a regular cough or having aches and pains or anything like that. So probably, I don't want to really give like I don't know that this is an accurate figure, but I would say like 90% of the time that we are sick, if not more, it's with a virus, not with bacteria, right? These are all really pretty serious infections. Pneumonia, bronchitis, it's like an infection that has kind of gotten worse over time, that isn't healing itself. But most of the time, we actually have a viral infection as opposed to one of these guys. Any questions about bacteria? Again, you probably know more about these than maybe about viruses and how they work, but it's still important to know about. Cool. Okay, so we'll jump into viruses then. All right. So viruses, we have this fancy technological term for them. We consider them biological entities. I'll get into why in a second, but we consider them biological entities. They can contain either DNA or RNA as their genetic material, and they require another cell in order to survive. More on that as well. So they have some, but not all the characteristics of life. And I have a checklist for you at the end of this that's going to highlight that. But most scientists agree, and there's still some debate about this, but most scientists agree that viruses are indeed not alive. They cannot replicate on their own, and it seems that they exist only to make more viruses. Okay, so they do have the nucleic acid, either DNA or RNA. There are some funky viruses out there. We have double-stranded DNA viruses, which would be like similar to our own genome. We have single-stranded DNA viruses. We have double-stranded and single-stranded RNA viruses, which is very unique, but all can be incredibly damaging and deadly. Okay, so as we click through some of the slides, I'll explain how they have some, but not all characteristics of life. Okay, so this is like a basic structure of a virus. They all have some sort of protein cap on the outside with nucleic acid on the inside, but that's about it. A lot of viruses will have some sort of protein marker on the surface, or glycoprotein, which would be like a sugar protein compound on the surface, and I'll tell you a little bit more about that in a second when we talk about the influenza flu virus. But they don't have ribosomes, so they're not capable of producing their own proteins. Okay, so this is the major reason why they are not considered alive, right? That means they don't have any enzymes. They don't have DNA polymerase or RNA polymerase. They can't make their own protein. They can't replicate their own nucleic acids because, again, they don't have DNA or RNA polymerase. They can't really make anything. They're just basically a shell with DNA or RNA inside. So they have to actually inject their genetic material into a host cell, and then the host actually replicates the DNA or RNA and makes the protein capsid for replication. It does all of that. Okay, so a little bit more into the structure, because these can look a number of different ways, and viruses are truly, I mean, you can major in virology and just study viruses for 4 years in college because they're so complex and so varied. But here are two examples that are pretty common. One is the bacteriophage, and this is a really important one to know about. It's involved in a lot of experiments, like early on experiments, and scientists are actually researching it right now as a possible solution to antibiotic resistance. So I can explain that in a second. But bacteriophages are viruses that infect bacteria. Okay, so they have this, they kind of look like moon landers, in my opinion, and they land onto cells and they inject their DNA or RNA into the cell, and then the capsid basically just disintegrates or disappears, right? So all that's left of that virus is the genetic material inside of the new host cell. Then the host, the bacteria, replicates the DNA or RNA as well as the protein coat. And makes copies of this kind of unknowingly. It basically hijacks the cell, and then eventually that virus will explode out of the bacterial cell, killing it and allowing it to infect other bacterial cells nearby. So scientists are looking to potentially program these to infect bacteria that we are infected with, because bacteria would only bind to viruses would only bind to certain host cells. They would infect and kill the bacteria, but not harm us. So that's in the works, but bacteriophages are really, really cool. And again, they show up in research a lot, so it's important to kind of know about them, and they just look really awesome. Kind of simpler and kind of like what we were looking at earlier would be the structure of the influenza or flu virus as well as HIV. So those have the protein coat, the DNA or RNA inside depending, and then they have these surface markers, those glycoproteins, or like a sugar protein combo. So if you ever watch the news and you hear about the flu epidemic, they will give the flu a name, the strain of the flu a name. So like this year, it's H1N1 that's flying around, okay? And that has to do with H and N glycoproteins that stick off of the surface of that viral cell. That can change from year after year. There's H3N2, there's all sorts of different strains that can come about. And when we talk about vaccines in the end here, I'll highlight how we use the different strains of influenza in the flu vaccine. But again, yeah, H1N1 is the one that's coming around. If you've ever wondered what the heck that means, it's just the naming of the different proteins on the surface of the virus. HIV also looks like that. We'll talk about HIV a little bit more in a second. HIV is an RNA virus, actually, but it has something really interesting called reverse transcriptase, which converts that RNA into DNA. Reverse transcriptase, transcription would be DNA into RNA, so it reverses that process so that the RNA can be converted into DNA and then implemented into the human cell. So it's a very strange inheritance there. Okay. So how do viruses work? So in order to replicate and make copies of itself, viruses need a host, right? Without a host, they are truly useless. Any cell can become a host, but bacteria, they're only going to pick a very specific cell to infect. So for instance, HIV only infects humans. It's human immunodeficiency virus, and it only infects human T cells, which is part of our immune system, which we'll talk about in 2 weeks. There are plant viruses, like the tobacco mosaic virus. There are pig viruses. Pigs have their own version of the flu. Dogs have their own version of the flu. But for the most part, you can't give the flu to your dog can't give you the flu, because viruses are going to be able to detect and infect only a particular host cell. And again, without the host, they can't function because they don't have their own ribosomes, so therefore they can't make any of their own enzymes or proteins. So the major steps that a virus takes, they attach themselves to the outside of the cell. They inject their genetic material into the cell. The host then replicates and assembles. So you can see the host is replicating and making the new protein capsids and new copies of the phage. This is an example of bacteriophage DNA. Those all get assembled and then they're released, right? And so we have all of these viruses then being released. The host cell dies, and all of them are then capable of going and infecting new bacterial cells. And you can see how rapidly this can kind of ramp up. So more examples of this. So what I just showed you was an example of the lytic cycle, and it's decently important to be familiar with these two cycles. So the lytic cycle is going to be where the virus just comes in, we're replicating the genome right away, we're making the proteins right away, it's busting out of the cell right away, and continuing. So this is when the virus is in like full-blown action. Now that happens a lot, but also on the same end, viruses can kind of go dormant, right? So a good example of this is the herpes virus, specifically the herpes virus that leads to cold sores. Okay, so 95% of Americans actually have this virus for cold sores, but obviously 95% of Americans are not walking around with cold sores every day. So what's happening then? Well, when you get infected, sometimes viruses will actually just kind of, again, go into this dormant mode where they just integrate themselves into the host. The same is true with HIV, right? HIV, oftentimes people are not aware for years to come because the DNA just integrates itself into the host. The host replicates as it normally would. Now we have a larger population of bacteria or human or whatever cells infected because the host is replicating and has truly no idea that there's like a strand of viral DNA mixed in with their own. This is continually happening until something happens that it would initiate the lytic cycle. So oftentimes this would be an event of stress or the immune system is suppressed. Perhaps you're sick with something else or stressed, or you're not sleeping or dehydrated. Usually when people are stressed, that's when a cold sore would appear. So the virus senses that stress, senses that the immune system is slightly suppressed, because that does happen when we're stressed, and it switches over into lytic cycle, full-blown lytic cycle, and that's where you see the effects, the inflammation, the swelling, the cell death that would lead to the cold sore. Again, same with HIV. That can lay dormant for a really long time until the body is under stress, and then it would switch into active mode, and those individuals would feel the symptoms of AIDS then. Yeah, it's the T cells, the T cells of the immune system that are infected there. So it has a large effect on the immune responses in people that do have HIV. Okay. Good with those two cycles. Anyone have any questions? Quiet group tonight. Hopefully you're learning things. Kind of interesting. I love this kind of stuff. Okay, so again, it is really important to know about those two things and our immune response to them, which we'll talk about in 2 weeks. Okay, so examples of diseases caused by virus. So a lot of things that you may think are caused by bacteria are actually caused by viruses. So again, common cold. Anytime you have a cold, aches, pains, flu, anything like that, it's a virus. Can't take an antibiotic for it. Don't take an antibiotic for it. More on that in a little bit. All of the hepatitis, A, B, and C. Herpes, all the types of herpes, mononucleosis, if you've heard of mono, which like totally knocks you out and infects your B cells, which are another part of your immune system, chickenpox, polio, influenza, measles, mumps, viral meningitis, AIDS, which is caused by HIV. So a couple things to point out, and we'll talk about them more when we talk about vaccines. Polio has been completely eradicated from the United States due to immunization. No one really gets chickenpox anymore for the same reason. Like back when my older sister was growing up, chickenpox was relatively common. When I was born, the chickenpox vaccine was pretty new, and so we all got that. And chickenpox, no one gets them anymore. Measles and mumps are two of the things that you're vaccinated for when you're very young, and those were pretty much eradicated until recently. There's a measles outbreak out in Seattle. because of a group of unvaccinated children. So that's kind of coming back. But again, a lot of these you may not have heard of or you've heard of, but know that there aren't any recent cases, and that's due to vaccination actually eradicating them so that they no longer really exist, except for in a vial somewhere in a top secret government building, I suppose. Okay, all right. So let's talk about our protection then, because obviously, well, our immune system protects us all day, every day. It's the unsung hero because you never really think about it unless it's not working, which means that it's usually working for you. And again, we'll get to that in 2 weeks and talk about how our immune system naturally keeps us safe. But we also have some assistance due to scientific breakthrough where we have antibiotics, antivirals, and then vaccination, which have led to a huge decrease in infection, in death from infection, all sorts of awesome things. So antibiotics, important thing with them, they can only be used to treat bacterial infections. So if you're going to the doctor and you have the flu or you have a cold and you get antibiotics, you are contributing to antibiotic resistance, which may take over the world one day, who knows? But they can only be used to treat bacterial infections. So unless you are really, 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 really sick and have been for a few days, you probably shouldn't be taking an antibiotic. How do they work? They target specific structures like peptidoglycan that are only on bacteria, hence why they wouldn't work against a virus, hence why they don't affect our cells. So they target very specific things that are common to bacteria. The problem with that is they often kill a lot of our good bacteria, especially in our intestine as well, which is why you might have diarrhea when you're taking an antibiotic, because a lot of your good bacteria that usually help you out have been killed. They're not smart enough to know the difference between a good and a bad bacteria. They just know bacteria. The original antibiotic, penicillin, which a lot of people are allergic to, although I just saw an article that 90% of individuals who think they're allergic to penicillin actually aren't. I guess better safe than sorry, though. That was made from a fungus. From that, most are now made artificially, but we've utilized the power of penicillin to penetrate the cell wall, and we have things like ampicillin, amoxicillin, all sorts of other cillins that have been derived from that, as well as a number of other antibiotics that have been made artificially that target specific bacterial structures. Antibiotic resistance, we have talked about a lot. So go back and watch lots of my old reviews when we talked about natural selection and tons of other things. Antibiotic resistance is a very real problem. You should look it up, read about it, keep yourself educated, because if you don't, who will? And it's something to be really familiar with because it's making these superbugs that are resistant to the things that we're trying to kill them with. Another way that you can help not contribute to antibiotic resistance is making sure that you finish your antibiotics whenever you get them. So like if you have a 10-day antibiotic and you feel great after day 6 and then you don't take the other 4 days of medicine, you're contributing to antibiotic resistance because there are most likely still some bacteria inside of you, and the bacteria that have survived the first 6 days already have some sort of resistance because they've survived for 6 days, even though you've been dousing them with chemicals. And so those are the strongest ones, and then they're able to replicate and spread because you haven't taken your full dose of antibiotics. Always take all of your antibiotics if you take them, and never take antibiotics for a virus. Okay, it's my public service announcements. All right, we also do have antivirals, which we never really talk about because they're not very commonly used. But they can only be used to treat certain viral infections. They don't kill the virus. They just kind of put them on freeze so that our immune system can kind of catch up and take over so that they're not replicating so frequently and kind of outrunning us. But they don't kill them. We don't know how to do that yet. I don't know that we ever will. Maybe we will. Maybe one of you guys will figure it out. So usually there's actually some complicated high risk kind of complications with this. So they're only given to patients who have very life-threatening symptoms or a life-threatening virus, who are hospitalized for it. There's actually some antiviral resistances out there as well. So they exist, they're cool to know about, but they're not very common and they're only given in life-threatening situations. Lastly, my personal favorite, vaccination. So here's what happens with a vaccine. Let's talk about the flu vaccine. So the flu vaccine has 3 different strains of the flu virus or the influenza virus in it. So basically, like scientists right now, they're predicting the strains that they think are most likely to show up during next year's flu season, right? So like a year out, maybe even more, I don't know. But they're making predictions. They're looking at trend reports. They're seeing what strains are popping up here and there. They're looking at traditional data and all sorts of stuff. And they're like, okay, these 3 strains are going to be the most likely to show up during next year's flu season. So those 3 strains are then gathered together and they're heat killed. So they kill them. Sometimes they weaken them. Sometimes they kill them. It depends on the vaccine. But they kill those strains. They put them into a serum, and then that serum gets injected into you. So when you get the flu vaccine, you have 3 dead virus strains inside of you. And basically, what your body does is they are then given time to figure out how to fight those 3 strains. So they analyze the virus. They come up with basically like a plan of attack, and I'm speaking very colloquially right now, not technically. I'll talk very technically about this when we talk about the immune system. But they come up with a plan of attack. They learn about these viruses, and then the next time that you are faced with that virus, so let's say one of your classmates has the flu and they sneeze and the virus enters your body already knows how to fight them. And the great thing about that is our body fights back like 100 times harder the second time they see something. And it's a really quick response because they already recognize the pathogen. And so you don't get sick. You don't even know that that flu virus was anywhere near you because your body was able to fight it off before it became a problem. So obviously we don't put living virus in those vaccines because you would get sick from living virus, right? But it's a really amazing, amazing way to prevent disease by giving you a weakened or a killed strain that your body can analyze and come up with a plan of attack for, and then remember for the next time you're exposed to it. Super cool. Love vaccines. We do have some vaccines against bacterial infections, but more commonly than not, they're against viruses. The reason you need to get vaccinated every year for like the flu is because the flu mutates very rapidly. And so if you had immunity to a different strain, you would not have immunity to the next strain. And then the other thing is sometimes scientists get it wrong. So like last year, the strain that was going around was not in the flu vaccine. This year it was. So H1N1 was in the vaccine. So if you got vaccinated, you are for the most part protected. If you were to be infected with it, you would have a much shorter and less severe case of the flu than someone who was not. Right? Pretty cool. All right. So this is our last slide. We're going to wrap up a little bit early tonight, but hopefully you found this informative. So this is kind of what I was referring to in the beginning of our time today, with the differences between viruses and living cells like bacteria. So viruses can have DNA or RNA at their center, whereas living cells would just have DNA, and they just have that and a protein coat, whereas we have cell membrane, cytoplasm, organelles, for bacteria, ribosomes, etc. For reproduction for viruses, they copy itself only inside of a host cell because they have no ribosomes, they have no enzymes to take care of that themselves. Again, they have DNA or RNA. They can't grow and develop. They can't obtain and use energy. They can't respond to their environment. And they cannot change over time. Okay, so that is a big portion of why viruses are, for the most part, considered non-living, whereas cells, bacteria, they can respond to their environment, change over time, obtain and use energy, grow and develop if they're multicellular, etc. So those are the major differences. That's a really important slide. And again, this will all be posted for you if you become a premium, I think we're calling it premium, premium member, or Fiveable Plus member, sorry, which again is just $20 until April 1st. So a great investment. You'll have access to all of my materials. I'll post worksheets and things up there for you, and be available to answer any questions that you might have. Any questions before? Oh, yeah, we have a question. Kristen, great question. So why do we call it heat killing then when we're inactivating a virus? I suppose because we don't necessarily have another name for it. I guess we could just say inactive as opposed to heat killed. Heat killed would be a better way to describe the process of getting rid of bacteria. Heat also does inactivate, I guess I should say, a virus. But I think heat killed just kind of stays because we are quote unquote killing them, making them inactive, but inactivating them is probably a better way to say that. Great question. Any other questions there? Cool. Well, viruses, bacteria. Hopefully you haven't had any firsthand experience with them this season, but I know it's hard. Know that going to school, you actually have a really amazing immune system protecting you because you're exposed to these kinds of things all the time. Got a lot of germy humans around you all the time because we're all germy humans. So keep up the good work. Get lots of rest. Stay warm wherever you are. I know it's cold pretty much everywhere, but stay warm. It was great talking to you tonight. Next week, we're going to talk about biotechnology. It's really, really cool. And then the week following, we'll talk about the immune system. And we're going to kind of jump into the immune system, the nervous system, and the endocrine system. So we have some really great weeks coming up. Tune in. I look forward to hearing from you. And make sure that you always reach out with questions if you have them. All right, guys, have a great night, and I'll see you next week.